Thank you for joining me today. My research project involves investigating genomic commonalities between different cancer pathway networks. In this research, we focus on searching for key genomic centers that are shared by multiple cancers and their interconnections. In the following demo, I will show you our preliminary results after visualization of all the 10 networks we worked with. On the left pane are all of the networks we have integrated so far. There are about 10 total. In our future work, we plan on incorporating all 23 of KEGG's cancer networks in our final cumulative merges. After forming the cumulative merge of all 10 networks, the result was this network, which has over 200 unique nodes and about 190 edges. As you can see, clearly it is quite messy and convoluted. After the application of our color scheme and formatting styles, this is the final network we are currently working on. The color scheme we created was produced by differentiating which genes were part of which networks and applying this information as a graphical overlay on all the containing nodes. In this context, a node is classified as a gene object and an edge is the line, or in this case, the arrow connecting the two genes. Each edge can express a multitude of processes. For instance, these two genes here are connected by this edge, which indicates that this gene activation will lead to the phosphorylation of this adjacent gene, which is further downstream. The data that we actually incorporated into Cytoscape is located down below in the table column. We were able to differentiate the network origin of each gene by uniquely tagging each network before the merge via commenting tags. After merging the networks, we discovered that several genes were key common centers for network interceptions. For instance, MAPK1, which is involved in the MAPK signaling pathway, was found in 90% of our networks, only missing basal cell carcinoma, abbreviated as BASO. In addition, our next most frequent gene, TP53, which is involved in p53 signaling, was also discovered to be very common, with it being found in all of our networks except renal cell carcinoma, abbreviated as RENA. The color scheme we created was very helpful in visually expressing which genes were part of one network. For example, MDM2 here is involved in 4 total cancers, these being glioma, which is marked by the yellow, melanoma, which is light blue, prostate cancer, which is dark blue, and bladder cancer, which is orange. Furthermore, this adjacent gene, FOS, is only involved in colorectal cancer, which is light green. To emphasize which genes were more frequent in all of our networks, we applied a sizing gradient. For example, the node for TP53 here is involved in 9 out of 10 of our networks, making it larger than most of the other nodes. Whereas the adjacent gene, CDKN1 alpha, is only involved in 4 of the networks, making it much smaller than TP53. By comment tagging each network before merging, this allowed us to use specific search commands to find the genes unique to each cancer network. For example, after typing this command, I can find the specific genes that are associated with this cancer. This is something which we look forward to using when enhancing our networks with JavaScript in the oncoming months. That was the end of the demo. Thank you for watching.